The topic that we'll be dealing about is the antigens. Antigens are nothing but the characteristic features of the organisms, and these organisms carry specific type of antigenic determinants that induces the production of antibodies when introduced into the body in a parenteral injection or in a parenteral way. So I am talking about this concept of parenteral is that when these antigens are introduced outside the intestinal tract, they are able to induce the production of antibodies. Otherwise, what happens is when the antigens are introduced via the intestinal tract or via the intestinal region, they are more getting degraded with the enzymes that are usually produced in the stomach or in the intestinal areas. So antigens are nothing but the characteristic features of the organism that induces the production of antibodies, which reacts with the antibodies in a specific and observable manner. And it is not that all antigens can induce the production of antibodies. There are some antigens that usually are able to sensitize lymphocytes and produce an immunological tolerance. Here the term immunological tolerance means the tolerance towards the antigens. So here there is no production of antibodies, but the antigens can be tolerated by the immune system. So all antigens need not be able to induce the immune status of the body to produce the antibodies. So antigens are nothing but the one that induces the production of antibodies, which reacts with the antibodies in a specific way and in an observable manner. The term specific way is used over here is because there is a specificity that is present between the antigen and the antibodies. So when an antigen is entering into the human body, it induces a production of lymphocytes. So these lymphocytes can either be a B lymphocyte or a T lymphocyte, and this lymphocyte carries specific markers that goes and interacts with the antigen. And in order to interact specifically with the antigen, it should have a complementarity between the antigen and antibody. Only then you can see it in an observable manner, or you can say there is a reaction between an antigen and antibody. And this is a basis that is mostly used in immunological diagnostic procedures, where the specificity between antigen and antibody is used to recognize a specific antigen pertaining to a particular organism. So there is a specific reaction. Specific reaction is only between the antigen and antibody, and it is a hallmark of an adaptive immune response where the antibodies or the T lymphocyte carry specific markers that can go and interact with only those antigens that are able to induce them. So it is a specific reaction that can be observed because there is a specificity and they get precipitated, which can be observed very clearly in vitro or in vivo. And it is not necessary that there is a specificity between the antigen and antibody. Sometimes a cross reaction can also occur. Now this cross reaction is nothing but when there are two different types of antigen carrying similar type of antigenic determinants, there is a possibility that they can induce the production of antibodies. And the antibodies that are produced, again one type of antigen, instead of going and reacting with it, it will go and react with the other. It is only based upon the antigenic similarity between the two types of organism. So in that case, a cross reaction can occur. Otherwise, the reaction between the antigen and antibody is so specific that they go and interact with them based upon the complementary shapes that are present on the markers or the surface of the cells. Either it can be on the B cell or the T cell and between the antigen that is present on the organism. And it is not the whole organism that can act as antigen. It is certain parts on the organism that usually act as antigenic determinants, or they are called as epitopes. So antigens are the one that induces the production of antibodies, or they can also sensitize and cause the immunological tolerance in our body. Now coming to the functions of antigenicity. So the concept of antigenicity or the important attributes of antigenicity is that it can either act as develop immune system or immune status called as immunogenicity, or it can also act in a way to react with the components of the immune system, which is called as immunological reactivity. So there is a difference over here. One is the antigen that is able to enhance your immune status to produce the antibodies, whereas in the second case there is no production of antibodies, but these antigens are able to react with the products that are formed during an immune reaction. So that is called as immunological reactivity. So based upon the function, you can classify the antigens into two types. One is called as a complex antigen, that is, or a complete antigen, that is able to induce the production of antibodies, and the other one are called as an incomplete antigen, which are also called as haptens. Haptens are incomplete antigen that are incapable of producing the antibodies. But there is one criteria where we can convert this hapten to make them immunogenic. That is, when you complex the haptens with the carrier, you can make the haptens into an immunogenic. That is, they are able to induce an immune response, that is to produce the lymphocytes or the antibodies. So based upon the function that they are either able to carry out an immunogenicity, or they are able to only react with the final product, that is immunological reaction, you can classify the antigens into complete antigen or an incomplete antigen, which is a hapten. Now haptens can be complex and hapten, or it can be a simple hapten. Now in case of a simple hapten, it is usually non-precipitating. It does not precipitate the antibodies, whereas in case of complex haptens, they are able to react with the antibodies and precipitate the antibodies. Now the characteristic features between these two is that the simple hapten is having only univalent property, whereas the complex haptens usually are polyvalent in nature, which are able to react with more than one type of antibody, and these can be precipitated. So here, based upon that, the haptens can be classified into a simple hapten, which is non-precipitating, and a complex hapten, which is usually a precipitating in nature. Now next, coming to the most important concept of antigen are the antigenic determinant sites that are present on an organism, which are called as epitopes. As already discussed, the whole organism will not act as an antigen. It has certain parts on the organism that are usually antigenic in nature, that is able to induce an immune response in the human body. So these are called as epitopes. Now these epitopes have specific property. They are specific chemical nature. They have certain groups that can go and interact with the human immune system to produce the antibodies, and they have a steric configuration that helps to develop the immune system. Not only they sensitize the immune system, but also they are able to react with the final products of the immune system, that is the B lymphocytes or the T lymphocyte, that usually reacts with them in a specific and observable manner. So epitopes are antigenic determinant sites that are present on the organism that induces the production of antibodies, and it has certain properties that helps to develop the immune system, and they are the smallest unit of antigenicity. That is, they are mostly made up of 4 to 5 amino acids, or they can be about a few polysaccharides that are usually present on the organism. And when these antigens are usually entering into the human body, they induces the production of antibodies, which carries specific markers that goes and interacts with the antigen, and these specific markers are nothing but the paratope. So epitope of an antigen reacts with the paratope of the antibody, and they usually are highly specific in nature. And these antigenic determinants or epitopes can be present in an organism in a sequential or in a linear segment, which are called as sequential epitopes, or they can be present in a quaternary structure of a protein, where the folding of the protein usually is the antigenic determinant site in a complete protein molecule, which is a functional protein molecule. So this is called as a conformational epitope. So based upon where these antigenic determinant sites are present in an organism, whether they are in a linear or in a folded form, they can be called as sequential epitopes, or they can be called as conformational epitopes. So the T cells will have the ability to recognize the sequential epitopes, whereas the B cell recognizes the conformational epitopes. Now the epitopes reacts with the paratopes to produce a specific reaction between them in an observable manner. But as already discussed, there are possibilities that cross reaction can take place when there are two different types of antigen having similar type of structures that induces the production of antibodies. So these antibodies may not recognize the original antigen, and instead goes and recognizes the other antigen. So this is called as cross reactions. So based upon the presence of epitopes, as discussed, they can be sequential epitopes or conformational epitopes. Now coming to the determinants of antigenicity of a particular antigen. So first property that has to to be satisfied by the antigen to be antigenic in nature is the size of the antigen. It is not that any antigen can induce a production of antibodies. There are certain criteria that are to be fulfilled by the antigen to make them antigenic in nature. So the first criteria that has to be fulfilled is the size of the antigenic molecule. The molecular size should be of very large so that it can induce the production of the lymphocytes in the body. So the high molecular weight compounds or the organisms or the antigens are able to produce the antibodies, whereas the low molecular weight compounds are non-immunogenic in nature. Whereas you can convert the low molecular weight antigens by combining them with the carriers such as bentonite or kaolin to make them immunogenic in nature. So as I already discussed, like haptens, we can combine the hapten with the carrier molecule to produce the antibodies or the lymphocytes. So the molecular size is very, very important to induce an immune response. So you should have a high molecular weight compound to induce the production of antibodies, and the low molecular weight can be converted by combining them with the large inert particles such as bentonite or kaolin to make them immunogenic in nature. So the molecular size is very important to make a compound or an antigen to be antigenic in nature to induce the production of the lymphocytes in the immune system. Now coming to the haptens, as I already discussed, they are low molecular weight immunogenic compounds that cannot induce the production of antibodies, but they can be made to convert into a complex molecule by combining them with the carrier to produce the immune reaction or to produce the immune cells in an human body. And sometimes the haptens can also produce an immune response in case of hypersensitivity. That is, a person who are atopic individual, in that case hapten can also act as a reagent that can cause an hypersensitivity reaction in an individual. Now next property that is to be satisfied with the for the antigen to be antigenic in nature is the chemical nature. The more complex the chemical nature of an antigen, the more antigenic it is. That is, there are different compounds with which the antigens are usually made up of. So the most potent inducer of an immune reaction is the protein and the polysaccharides. So they rank first to act as antigenic in nature. So protein comes first, then comes the polysaccharide. Potent antigenic character of an organism that induces the production of lymphocytes. Next, coming to lipids and nucleic acid, they are said to be low immunogenic, and when we want to produce antibodies against these lipids and nucleic acid, we have to complex them with either the proteins or the polysaccharide to make them immunogenic in nature. And chemical complexity is very, very important. The more complex the chemical is, the more antigenic in nature. So if the structure of the particular compound or the antigen is simple, then it is low immunogenic. If it is more complex, then it can be more antigenic. For example, in a protein molecule, there is a quaternary conformation of the molecule that is functional in nature, and this quaternary conformation of the protein is more immunogenic when compared to that of the polysaccharides that are usually made up of only 4 to 5 glucose or the carbohydrate molecule. So the more complex the structure is, the more immunogenic in nature. And apart from that, there are certain proteins which are usually non-immunogenic in nature. Take for example gelatin, because it has a shape that keeps on changing. So this gelatin cannot be immunogenic in nature. So chemical nature, the more complex the chemical nature, the more antigenic in nature. Now coming to the third important aspect, that is the susceptibility to tissue enzyme. The more easily these antigen gets processed, the more antigenic they are. But here you should make it a point that the more rapid susceptibility to tissue enzymes are not antigenic in nature. So more easily processed, the more antigenic in nature. Now these antigens are usually processed by the phagocytes in our body. So once the phagocytic cells or the macrophages have processed this antigen, they are now able to act as immunogenic component in the body. So highly processed antigens can be more immunogenic in nature, and when these phagocytosis is taking place, the antigen is degraded into particular fragments of variable lengths. Now these fragments are usually the one that induces the immune system. So easily processed antigens are highly immunogenic in nature, and they are processed either by the phagocytes or the intracellular enzymes of the immune system. And next coming to polystyrene type of material, they are non-immunogenic in nature, and the substances which are made up of D amino acids are non-immunogenic in nature, but the substances which are made up of L amino acids are highly immunogenic in nature. So the simple thing to say about this is, the more easily antigen gets processed, the more antigenic in nature, and highly processed antigens are more immunogenic in nature. Next property is the foreignness. Foreignness is, it should be completely non-self to the human body. So the degree of foreignness will tell you the degree of antigenic property of that particular antigen. The more like it is unrelated to the human body, the more antigenic in nature. Now take for example, bovines are closely related to the humans. Now the antigen from the bovines, when introduced into the human, they are little low immunogenic in nature, whereas the antigen introduced from some other species can be more immunogenic in nature. So the antigen should be completely non-self. Now the logical reason behind this is that the self antigens are usually exposed during the birth, that is during the development of the fetus, to the lymphocytes. As a result of which, the lymphocytes become tolerant towards the self antigen. So that concept is called as immunological tolerance. So because of the immunological tolerance, we don't see that the self antigen being immunogenic in nature. So it is the non-self antigen or completely foreign antigen that is highly immunogenic in nature. Now when this immunological tolerance usually breaks down, this leads to autoimmune diseases. Now the concept of autoimmune diseases is that our self antigens, there are different antigens present in our body. So these antigens have been exposed to the lymphocytes during the development of the fetus, and they are told that these are the self antigens, so no immune reactions has to be produced against our self antigen. Now once this control is broken down in the later part of the life, it will lead to autoimmune diseases, which was proposed by Herlix, who said that a concept of horror autotoxicus, that is poisoning of the self antigen when there is a breakdown of the immunological regulation in our body, where the self antigens are detected as non-self, and they are usually acted upon by the lymphocytes of our own body to lead to autoimmune diseases. So the more degree of foreignness, the more unrelated the antigen is, the more immunogenic in nature. Now next coming to the antigenic specificity, which is an important property of the antigen. So the antigenic specificity is said to be stereochemical in nature, and it is determined either by the single chemical grouping or a single acid radical, and the position where these components are usually present, whether they are present in the ortho, meta, or any other position in a particular antigen, determine the specificity of that particular antigen. Apart from that, the spatial conformation, whether they are levorotatory, dextrorotatory or mesorotatory. Again, among these also there is a difference in the specificity of that particular antigen or antigenic determinant, which is the epitopes. As already discussed, when there is a structural similarity of the antigen on different types of organism, there can lead to a cross reaction. Now there are two aspects of this cross reaction. One is a beneficial effect and the other one is the harmful effect. Now coming to the beneficial effect of the cross reaction is the immunization with the cowpox to save us from the smallpox virus. Now the disadvantage of this cross reaction is that the human heart contains certain types of antigen which are similar to the M proteins present on the streptococcal organism. Now during an infection of the streptococcus, the antigens the antibodies that are produced in our body, instead of going and reacting with the M protein of the streptococcus, they go and react with the heart protein, thereby damaging our heart. So this is a disadvantage of a cross reaction. But ultimately, there's sometimes the specificity is not very perfect, that it might lead to a cross reaction. Now depending depending upon the tissue antigens, you can classify in an animal, you can classify the antigenic specificity into 4 types. One is called as the species specificity, isospecificity, autospecificity and organ specificity. Coming to the species specificity, it is pertaining to particular species, that is the tissue antigen of all the individual in an like a species shares the same species specific antigenicity. Now this based upon this immunological reaction, you can draw out the phylogenetic relationship between the different types of species. So it helps to trace the evolutionary relationship between the different types of species based upon the species specific antigenic property. So the isoantigens helps us to classify the person in a particular group and helps us to group them in particular species. So here we have isoantigen example, the human erythrocyte antigen. Based upon that, you can classify the different blood groups and you can group the individual based upon the blood groups, and these are usually genetically determined, and it is very helpful when you are going for a blood transfusion. From when you are transfusing the blood from one person to the other person, the compatibility is very essential. There is a person with the A blood group can give the blood group only to the A person but not to the B. So this isoantigen help us to group the person based upon the antigens that are present in a particular species, some members of a species but not all the members of a particular species. Also this isospecificity is helpful in isoimmunization of during the pregnancy, and we have the other important specificity is the autospecificity. Autospecificity is nothing but the presence of autologous or self-antigen. Mostly the self-antigens are non-immunogenic in nature, but sometimes there are chances that these self-antigen can become immunogenic. For example, the sequestered antigen present in the eye lens that is hidden in a capsule. So when there is an accident, when there is a wound to this eye lens, these are exposed as a non-self antigen and can develop an immune reaction. Apart from that, the production of sperms in the later part of the life, and these sperms are not exposed to the lymphocyte during the exposure to the fetus, so they are usually taken as non-self antigen but they are not immunogenic in nature. So autospecificity or self antigens are non-immunogenic in nature, except in certain cases where there is an exposure of the eye lens protein during a particular damage, they can become immunogenic in nature. Next is the organ specificity. Organ specificity is nothing but there are certain organs such as brain, kidney and lens protein which share the similar antigen with different types of species in an environment. So these are the antigens which are present in different types of species present in a particular organ is called as the organ specificity or the organ specific antigen. Next is heterogenic or heterophilic antigen or specificity antigen. Here we have the closely related antigens that can be shared between not only the different species but also between the different kingdoms, different classes, etc. So best example is a phosphine antigen which is present in animals, birds, plants, etc. And it is the same type of antigen which is a lipid carbohydrate present in different classes, different kingdom and different species of a particular environment. It is not present in the rabbit, and here we can use this rabbit to produce the antibodies against the phosphine antigen. And apart from that, these heterophilic antigens are mostly used in serological diagnostic tests in the immunological assays. So now coming to the biological classes of antigen. So now the immunogenicity of a particular antigen is when the antigen enters into the body, it is able to produce the immune response in such a way that they are able to produce the lymphocytes such as B lymphocytes and T lymphocytes to go and interact with the antigen. Now the antigen, there are certain antigens that can directly produce the B lymphocyte without the involvement of the T lymphocyte, whereas the involvement of T lymphocyte is very essential for the production of an immune response. It is a T lymphocyte that produces the cytokines that helps to activate the other cells of the immune system. But there are certain antigens that can directly go and produce the B lymphocyte without taking the help of a T lymphocyte. So depending upon whether they are going to directly activate the B lymphocyte or involves the presence of T lymphocyte, you can classify them into two major classes. One is called as a T cell independent antigens and T cell dependent antigen. The name itself says that in T cell independent antigen they are directly able to produce the B lymphocyte without any help of T lymphocyte, whereas T cell dependent activation requires the involvement of a T cell that produces cytokines to produce the B cells or the antibodies from the B cells. So now the characteristic features of this T cell independent and T cell dependent is that the T cell independent are very simple in nature, simple antigens in nature, and there is an immunological dosage that helps to activate the immune system to produce the B lymphocytes. So it is called as dose dependent. It is not only one or two organisms that can activate the immune system, but there should be certain groups of organism or certain types of organism that has to enter to develop the immune response. That is called as a dose dependent. And low dose will be low immunogenic, and high dose will become immunological tolerant towards a particular antigen. So a certain amount of dose, a minimum dose is required to activate the immune system. Next is the antibody response towards this particular antigen, which are T cell independent, is majorly via the IgM and IgG3 immunoglobulins, which are nothing but the classes of antibodies that responds towards the antigen that has that is activating the immune system via the T cell independent mechanism. And then here in this particular mechanism you will not find immunological memory, that is there is no memory response. Now when the antigen enters into the body, two types of responses are produced. One is called as the effector response, the other one is called as the memory response. The effector response is the primary response towards that particular antigen, whereas the memory response is the secondary response when on a subsequent infection with the same type of organism, the memory cells are present to interact with that particular antigen. So here in the case of T cell independent antigen, there is no immunological memory, no memory response, and here there is no requirement of any processing by the macrophages, whereas for the T cell dependent you need processing via the macrophages, because after the processing the antigenic fragments are exposed on the major histocompatibility complex that are going and interacting with the T cell receptor. So T cell cannot recognize antigen directly. It requires a cell in between which is called as the antigen presenting cell that presents the antigen to the T cell. Only then it will get activated to produce the cytokines to activate the other cells of the immune system. Now T cell dependent, it is more complex. The antigen should be complex, and it is immunogenic in nature, where it helps to activate both B cells and T cell via the T lymphocytes. And then the antibody response towards the T cell dependent are the major classes of antibodies such as IgM, IgG, IgA and IgE. So all the groups of the antibodies are involved in case of T cell dependent antigens. And then you have a immunological memory that can be seen in case of T cell dependent antigen, which is absent in T cell independent. And processing, as discussed, only after the processing by the macrophages the antigens are presented to the T cell, and it is only via the processing or the phagocytosis that the T cells can be activated. And finally the antigen will be metabolized completely in the phagocytosis to get exposed onto the major histocompatibility complex to activate the T cell. So these are the biological classes of antigens. So summarizing the antigens, it is nothing but they are the substances that can induce the production of antibodies when injected parenterally, and they are the one that reacts specifically with the major components of the immune system, that is the B lymphocytes and T lymphocytes. And based upon this, we are able to develop various immunological diagnostic procedures to identify the antigen or antibody related to a particular organism.